all our panelists. I think it's really interesting time to be in data analytics and data science. So we are going to start session 3 now and resume our talks from 3 other topics that are basically going back to COVID-19 and again trying to think about how we can deal with this pandemic. So our first speaker is Crystal Hepp from Northern Arizona University, and she is going to talk to us about mechanisms to find early warning signs of COVID-19 outbreaks. Crystal, you should be able to share your slides and unmute yourself.
Crystal, we can see your screen, but you're muted right now. You'll need to unmute. There you go.
Can you hear me now?
And if you'd like to turn your camera on, you're welcome to. Yeah, we can hear you. Thank you.
Okay. I think I'll keep my camera off just to preserve bandwidth at my home. So thank you for having me today. As Fabio said, my name is Crystal Hepp, and I'm an assistant professor at the School of Informatics, Computing, and Cyber Systems at Northern Arizona University, and also an assistant director of the Pathogen and Microbiome Institute. And today, I'll be talking with you about wastewater testing that we're doing in Northern Arizona. My background over the past few years has really been focused on West Nile virus and St. Louis encephalitis virus circulation in the southwestern United States. However, we did submit a grant about 3 years back to look for norovirus in wastewater with the idea that if we could identify it in the wastewater prior to it spiking up in the hospitals, that we might be able to use wastewater as an early warning system. And now a lot of groups during the COVID-19 outbreak, they have also taken to this idea of using wastewater as an early warning system. So the first thing that we did back in March is we reached out to several of the communities in Northern Arizona. So you can see the encircled area here is the area that we're really focusing on. And we're working with the communities of Munns Park, Kachina Village, Sedona, the village of Oak Creek, which is also referred to as Big Park, and it's just south of Sedona. And then finally Flagstaff, which is the largest urban area in Coconino County and also in Northern Arizona. And what we're doing is when we work with wastewater treatment plants, we are collecting composite samples once weekly. And so a composite sample is a collection of 15 milliliters every 30 minutes over a 24-hour period. And then from that total amount, we collect 4 different vials of 40 milliliters for downstream processing. However, what we found out when we switched to being interested in doing college dormitory sampling is that the college only had 2 composite samplers, and to cover all of the different dormitory sites 3 times weekly, we were not able to move those composite samplers around quickly enough. So what we did is we looked back into the literature and found that in the 1940s, there was the development of the Moore swab, which is essentially a 4-foot piece of gauze that is wrapped up and then placed into a manhole for 24 to 48 hours. And we still had issues with that because it takes time to wrap up those swabs, and when you're touching them, they become contaminated with your own flora, and we didn't want to waste time putting these through the autoclave. So instead, we decided to start using tampons. So what we use outside of the dormitories is we take an OB tampon that doesn't have the applicator. We tie a string to it, we dip it into the manhole, and we leave it there for 48 hours. We come back, and then we put it through a downstream processing protocol in our laboratory. That protocol includes viral concentration because the virus is quite dilute in these water samples. So it undergoes a process called skim milk flocculation to concentrate the virus. We follow that up with both a DNA and RNA extraction using Trizol. The reason that we are looking for DNA as well is so that we can look for additional viruses that might be present in the wastewater. For the RNA viruses, we have to put them through the reverse transcription process. We use the CDC N1 and N2 assays in a quantitative PCR to estimate the total viral load in every one of the samples. And just recently, we've started using the Swift Biosciences child amplification procedure. What that is, you can look at the figure at the bottom, and that involves taking primers and scaffolding them across the entirety of the 30,000 base pair SARS-CoV-2 genome to be able to get a final genome that we can place together bioinformatically. And then finally, we perform our sequencing on one of our aluminum ICs at the Pathogen and Microbiome Institute or the Translational Genomics Research Institute. These are the qPCR results over time for the city of Flagstaff and also the case count. And the bars represent the viral signal, which is also on the first y-axis at the wastewater treatment plants. And then the black line represents the case count over time in Flagstaff, so where the wastewater treatment plants are catching the samples from. What you can see is that the wastewater results, the signal, correlates fairly well with the case count over time in Flagstaff. And you can also see that when the cases were spiking in Flagstaff was about the same time where the Flagstaff mask mandate was implemented, and the case count went down for several epidemiological weeks over time and has just recently started spiking up again. So what we've done with some of the samples is we put them through the Swift Biosciences child amplicon sequencing procedure. And most of these samples have a CT value ranging from 37 to 42, which is quite high. It's a poor signal for performing sequencing on, but we tried to anyways. And you can see from this read pileup that I'm showing that we did end up sequencing a good portion of the genome. We're trying to improve some of the primers to get better amplifications for better downstream sequencing. But something that was very obvious and that we were expecting is that we would get strain mixtures that were resulting from this sequencing procedure. And that's not surprising because what we're doing is we are first chopping in the genome into several different amplicons and then placing it back together. And from this region, this blown up region, what you can see is that we have several mixtures that are the majority or the minor allele frequency is quite high at several of these different sites. And if we were to use our typical consensus sequencing, our consensus sequence calling protocol, what we would find is that all of these mixture sites would be called ends instead of actually recognizing the diversity that's present in these samples. So moving forward, we haven't done this yet, but what we're planning on doing is using a 100% OTU clustering protocol. So where we take all of the reads that are identical to each other and we give them their own sequence name, and then we're going to use a program to place every one of the read clusters onto a phylogenetic tree so that we can actually capture the full diversity of SARS-CoV-2 that's present in the wastewater sample at any given time. However, we have moved forward, and so this wildcat sample that you can see in this figure in the red, the figure that's on the right, that we did place that into a phylogeny. This phylogeny on the left is from a paper that we recently published. And when we took our wastewater sample, we did find that the May 8th sample, which was the first one that was, which was the first positive sample in Flagstaff, we find that it clustered with the sample from Maricopa County, which is where Phoenix is located. So we're still working on this, but we do think that it's possible that this is that this particular strain was an importation from the Maricopa County area. And what I wanted to briefly discuss was another project that we're working on that we're trying to model the wastewater project after. So we have a special community in northern Arizona that we're working with. And in that community, they've done extensive, it's a small community, only a few hundred people live there. And we've done extensive genome sequencing of all of the positive cases. And that's what is shown here in this phylogeny. And then above, you can see the sky grid, which represents the affected population of the virus over time in that particular community. And then what we did is we compared the effective population size to the number of cases in this very well sampled and well monitored community. And we found that the effective population size correlated quite well. So you can see 86% of the diversity in cases can be explained by effective population size. And in fact, what we later found is that there were 2 different lineages of SARS-CoV-2 circulating. The phylogeny only represents 1 lineage. And when we combine the effective population size estimates for both of the 2 lineages, they even better explain case count. So we get about 91% of the case count being explained by effective population size. So what we're trying to do with the wastewater project is move forward so that we can use wastewater samples in the absence of really good human testing so that we can predict the outbreak magnitude over time in these different communities. I'd like to acknowledge all of the different groups that we're working with. We're working with City of Sedona, Oak Creek, Flagstaff, Munns Park, which is the Pinewood Sanitary District, and then Kachina Village Improvement District. We've got several collaborators at Northern Arizona University that are working on this, and we're also collaborating with the Translational Genomics Research Institute on this project. Thank you.
Wonderful. Thank you, Crystal. So I forgot to mention before, if anybody has questions, feel free to type them out in the Q&A or in the chat so that I can read them out. I do have a couple of questions for you, Crystal, just to start things off. The connection that you see between cases and effective population size, do you see any particular connection with specific strains, or you simply haven't been able to test it because you've done it only on 1 sample? I'm just trying to get to, are there certain strains that are going to be easier to identify with this method compared to others?
Let's see. Sorry, what do you mean as far as strains that are easier to identify?
So if you are using wastewater as a way to identify, to predict outbreaks, is it possible that we may miss potential outbreaks because some strains can be identified in the wastewater and others cannot?
Well, I don't think that it's a matter of strains that can versus can't be based on the genomic sequence. Because these primers have worked very well on a variety of different strains. However, if there is a very low-level outbreak, it is possible that we will miss these strains if we're sampling at if we sample at the city level. However, with the congregate living sites and even in very smaller cities, we do find that we are able to identify spikes in the population prior to case spikes. So we do think that we're able to identify very low-level strains. So I don't think that that's necessarily an issue. Maybe if there's only 1 or 2 people in the community that are infected with a particular strain, that could be more difficult.
Right. And I know that this work kind of you have worked on other infectious diseases before that behave quite differently from COVID-19. I'm just curious as to how did you transition from the work that you did before to the work that you're doing now, and what kind of overlaps do you see between all the different types of infectious disease analysis you did?
So it was quite easy for this particular example because West Nile and St. Louis encephalitis virus are both RNA viruses. They're shorter than SARS-CoV-2, but they're RNA viruses, so the sample processing and the downstream sequencing protocols are very similar. The only thing that really differs are the primers that we use to amplify these different viruses. So I think that when you're working with a particular type of virus, it's pretty easy to transition if you're already within that area. The only difficulty that we had was the actual sample collection, so we had to learn how to do that. That took us about 2 weeks to get up and running.
well, that's not actually that much to learn how to do a brand new sample collection. And I like that, you know, what they say, the necessity is the mother of invention. And so you found a good way of doing your sample collection. Okay.
Another question for Dr. Hepp. Dr. Hepp, could you talk a little bit about the size of the data that you are dealing with, you know, and what kind of tools that you use? If you could comment on that a little bit. Thank you.
Sure. It really depends on the project. So I'm involved in several of the different COVID projects in Arizona. For the larger project, what we, you know, now there are, I think, more than 40,000 publicly available SARS-CoV-2 genomes. And so one of my collaborators, Greg Caporaso, who's also the head developer of the CHIME2 package, he developed a genome sampler, essentially, that will take all 40,000 strains, for example, and downsample them to a representative set we've run up to 2,000 genomes on our high-performance computing cluster. And so without the high-performance computing cluster, we wouldn't be able to process these genomes quickly. But essentially what we do is we use an alignment program through IQ3. And then we are performing the phylogenetic analyses using BEAST with the high-performance computing cluster. And that's what we're doing.
Thank you.
Excellent. Any other questions that I may not be seeing in the chat or the Q&A? I don't think so. Okay. All right, that sounds good. We are a few minutes early, but that will give us a little bit of time to make sure that technology collaborates. So I think we can switch. Thank you, Crystal, for showing us your very interesting project, and hopefully we'll, you know, be able to use these early warning methods kind of more nationwide to keep all the various outbreaks that are happening under control, especially related to campuses as we have all reopened. Okay, then I think we can move forward with our next speaker.